Ma'am's current position is principal consultant. Ma'am's current position is principal consultant and scientist in Chitale Dairy, Sangli, Maharashtra. And ma'am is also consultant microbiologist in one of the local dairies in Ontario, Canada. Ma'am has served 24 years as a professor in RKT College, Thane. And out of that, 8 years where ma'am was HOD, microbiology. And ma'am was in the college, University of Pune, and ma'am was HOD Biotechnology for 5 years. Afterwards, ma'am has also served as a principal in Lokmangal Biotech College in Solapur University, Maharashtra. And other than that, ma'am is also a visiting faculty, assistant professor for Singer College of Engineering and SSP University of Pune. Ma'am has also served as a consultant in Amphenol Interconnect Private Limited, Pune, for 4 years. And there are lots and lots of research papers, publications, international journals, and lots of international presentations. Ma'am has also served as poster presentation judge in various events. Ma'am has currently on board peer reviewer of respected Elsevier publications, and she is in the journal Food Science and Technology. Ma'am is also an advisory board member in International Journal of Biotech Applications. Ma'am is also life member of lots and lots of different scientific organizations, and she is very active. So in total, I would like to conclude that ma'am has more than 4 decades of rich experience. And now I would like to invite Dr. Sarita Mahajani to start her presentation.
A very good evening to all, to one and all. Thank you, Jay.
Shilpa ma'am, can you speak 2 minutes? Shilpa ma'am, can you unmute yourself? If there is a problem, Sarita ma'am, you can continue.
Yeah.
Yes. Shilpa ma'am.
Yes, sir.
Yes. Now you can speak for 2 minutes. Welcome from Gujarat in chapter of Microbiology Society.
Thank you. Thank you so much, sir.
Hello. Shilpa ma'am, you speak for 2 minutes.
I think there might be some issue. She is not able to unmute.
Okay. Sarita ma'am, please continue.
Okay. Hello to everybody. Very good evening to one and all. Thank you, Jay, for the elaborate introduction. I hope all of you are keeping safe. I am very grateful to the Microbiologists Society India, the President, Honorable Dr. Deshmukh sir, the Convener, Professor Shilpa Madam, President, Gujarat Chapter, and the Coordinator, Research Scientist, Mr. Jay. Thank you so much. It's a pleasure to be here with you all today, and I appreciate being given the opportunity to speak. It's nearly the end of the day there, while it is bright winter morning, beautiful morning here, 7:15 a.m. and it's a beautiful winter. It's nearly the start of a fresh day. We are 2 months now in 2021. We had 2022, in which the pandemic really became a crisis in all ways and forms of life. I hope 2021 is much, much better. The pandemic will slow down. The coronavirus, as you have seen, is no match to science, but we must finish the job. This was a report given on 4th March 2020 by the New York Times. More than 90,000 individuals across the globe that time had been infected by the novel coronavirus, and the number continues to climb. That was the report given. Since the coronavirus was first detected, the bioindustry, dedicated scientists, and researchers from more than a dozen companies stepped forward to develop vaccines and antiviral therapies to contain the quickly spreading viral illness. The science is galloping, they said, forward, and it is vital to translate this science into solutions to combat the virus. In the 21st century, as we have seen now, knowledge and know-how are the keys to success. So talking about biotechnology is not a new thing now. It's a broad field. In particularly, I'm going to discuss the biotechnologies used in industries, the fascinating, inspiring new techniques used, the principles underlying these technologies, their applications to practical, and we will start the first session today by the most recent technique that is used by all the industries, that is the field of vaccinology. So it's not going to be a textbook talk in these 3 sessions. We'll start with vaccinology today, understanding the industries, the candidates, the vaccine candidates. Next session, next week, will be the production of vaccines by these industries. And the third session will be general with all the other biotechnologies that are recently used by the industries. So what will microbiology be like in the world where fixing your body and fixing your computer become the same thing? So today, let us start with, to reach Mars, we need biotechnology. So it's a technological application of biological organism systems and processes to develop, make, or modify products to specify uses. Although the definition is certainly no soundbite, it points out 2 important things, namely, biotech is a series of techniques, and biotech by definition focuses on products and practical applications of science, not science for the science's sake, of course. It is the technology that uses biology and functions of living microorganisms to make new treatments and technology for healthcare. It is advancing and stepping stones to human progress. It has the areas which you can say genetics, DNA sequencing, recombinant gene techniques, applied immunology, diagnostics. And if you speak about applications, you can say biopharmacy, bioagriculture, bioindustrial, bioinformatics, etc. So biotech companies derive their products from their extraction or manipulation of living microorganisms. And to compare it with pharmaceutical companies, which create medicines from chemicals and synthetic processes. In the world of investing, these both are important industry sectors with very different risk profiles, despite their overarching similarities. So as human beings, we need a constant supply of oxygen. We need constant access to nutritious food, clean water, and a safe and clean waste disposal system. This was said by Italian astronaut Samantha in 2015, 30th April, a live stream from International Space Station. So she says that establishing a base on the moon or taking humans to Mars for the first time is the goal of several countries around the world. To be able to survive a long-term mission, you need to have transport to a lot of water, a lot of food, and a lot of oxygen. And to prepare for long-distance space travel and life on Mars, biotechnology will be essential to make spaceships no longer dependent on Earth's constant supply. So going forward now, I would like to take, before we start the session, proper session, I would like to take a couple of minutes to check on the COVID life statistics, the recovery awareness, and then the reference sequences. This is the site called as Worldometer. Of course, there are many trackers. There is CDC tracker of cases as well as the deaths. There is John Hopkins tracker, but I'm more comfortable with this Worldometer, which manually analyzes, validates, and aggregates data from thousands of sources in real time and provides global COVID-19 live statistics for a wide audience of caring people around the world. Worldometer is a site that calculates the estimated population, coronavirus and a wide variety of statistics in real time. How do they work? They collect data and process it around the clock, 24 hours per day and 7 days per week. Multiple updates per minute are performed on the average by their team of analysts and researchers who validate the data from the ever-growing list of over 5,000 sources under the constant solicitations of the users who alert them, as well as official announcements, and is made around the world. So if we get time today at the end of the lecture, we will go properly to this particular site and find out today's case analysis, what it says. So moving on to the next one, this is one more. It's called as Long Hauler Science Tracker. Many patients with mild to moderate COVID-19 become long haulers, suffering symptoms months after they clear their non-life threatening infection. Researchers have found about 33% of the COVID-19 patients who were never sick enough to require hospitalization continue to complain months later of symptoms like fatigue, loss of smell or taste, or even the brain fog. If you contract coronavirus, there is a good chance you could experience a lingering effect. And this is the site which is created as a resource called as C19RecoveryAwareness.com. The research and studies of the long-term effects, mild or moderate, on the case recovery. It's around internationally, it is called as long COVID, and with patients here referred in US, it's called as long haulers. The need for continued awareness, research, and rehabilitation is becoming all too important as a number of cases grows, and can be found here at on this particular site. So we can even go through this site and find out how it works. Weeks back, there was a news saying the virus is still winning, and what else you need to know about. The chart here on the left-hand side compares the spread of virus in each of these two countries with the spread in a group of nearby countries. As you can see here, the cases surged in Britain and South Africa since the variants first surfaced, while holding fairly steady in rest of the Western Europe and Southern Africa. This report suggested the rest of the world may now be at risk of a COVID-19 surge. The Atlantic paper also reported it was a mutant ninja. On the left-hand side below, you can see, I have highlighted it, mutant ninja. And this data on a new variant rolled in, and there came the cause of the real concern actually. The variants already seemed to have spread around much of the world. More than 30,000 countries have diagnosed cases with the variant first detected in Britain, and it's known as B117. Let's not call it as UK variant. And scientists said that it could soon become the dominant form of the virus. There was a report on 20th September, however, and then in December, it became the dominant strain in London also. This was a new strain of SARS-CoV-2, which had spread, and it was named for the first time as VUI 2020-12/1, the first variant under investigation in December 2020. The emergence of genomic diversity and recurrent mutations in SARS-CoV-2, if you read the highlights right in the center, 198 sites in SARS-CoV-2 genome appear to have already undergone recurrent independent mutations. Now, the mutations that are focused on have emerged independently multiple times, and 198, which is recorded here, recurrent mutations in the SARS-CoV-2 genome was identified. Nearly 80% of the recurrent mutations produced non-synonymous changes at the protein level, suggesting possible ongoing adaptation of the virus. Now, moving on, this is a site where we can find out the mutations. SARS-CoV-2 is a SARS-like coronavirus, and it's of zoonotic origin, acquired from animals, first identified in December 2019 in Wuhan, China. The virus has since spread globally, resulting in a currently ongoing pandemic. The first whole genome sequence was published on January 5th, 2020, and thousands of genomes have been sequenced since this date. Virus tends to mutate. The virus is new to us, but they are trying to adapt to the new environment, that is the host environment, changing some of their proteins every time they divide. So the mutations possible? Yes. Are the mutations happening? Yes. Are there some strains of viruses? Are these multiple strains? Are they lethal than the other strains? So tracking the strains is important for the scientists as well as the whole world in science. So there is the site called as nextstrain.org. Nextstrain SARS-CoV-2 resources around the world. People are sequencing this particular virus. The Nextstrain team analyzes these data on the global and continual level. It's a continental level, of course, continuously done. More specific analysis are often provided by the groups around the world. And this page lists all the SARS-CoV-2 analysis that use Nextstrain that we are aware of. They have phylogenetic region-wise of the virus strains, the transmission, diversity, geography, frequency. You can see all of the strains are still evolving and they are cross-contaminating as people are traveling from one place to another. So you can have even the clades there, a group of organisms believed to have evolved from the same ancestors. So moving on, let us see how this works. I'll take you to the site. Can you see the next page? So here you can see it started here at this particular point, Wuhan, right? And this is the first time it was found out. And if you hover your mouse, you go on here, India, Telangana. It is from CCMB. If you click here, the mouse, you can find the details of it, the collection data, the authors who have submitted this, the title of the paper, published paper that has been given, then the genomic studies, the comparing the studies. If you take the strain, if you find out the country, the clade, the age of the patient, then you can even find out the host, location, the originating lab, submission date, region, sex, and emerging clade, which is 19A. And this has been submitted by Center for Cellular and Molecular Biology, CCMB. So if you check out here, yes, if you check out here, you will find they show here, oh, sorry. So you can see here the nucleotide sequence mutation, the amino acid mutation, all of them together. If you click on one, you can find here nucleotide sequencing. You can find the mutations here. Let us go back to, the Indian strain here. Here you can find it is from Telangana. It will tell you the nucleotide mutation, the amino acid mutation as well. The amino acid mutation means that it has 3 nucleotides, the mutation together done, formed. And the letters stand here for the nucleotide bases that comprise DNA, that is thymine, adenine, guanine, and cytosine. Looking at the location here at this particular strain, you will find it has mutated at this location site 1820 on the chromosome. If you see that G is the normal value, that is guanine, and the mutated value is adenine, that is A. And this is how the mutation is seen here. Then you have the amino acid mutation here, open reading frame A, 1A or 3. 1A is here, G519 location here. So these are the mutations that you can see, and you can also see how the divergent, how much divergent it is. You can see the date, the change, the country, the city. It's about nearly 3,000. If you move on, if you hover your mouse here up till end, you will find here at the end. These are the latest ones in February end, or you can say March. All the variants that you can read here, and these colors are for the different clades that are seen here. And this site says that the visualization can handle only around 3,000 genomes in a single view for performance and legibility. Okay, now coming back to the okay, I think I have missed out my presentation. Let me start back again. Oh, I'm sorry, I just missed out the presentation. Yeah, can you see now?
Yes, ma'am.
Yes, again, first slide is there.
Yeah, just a second. Yeah.
Correct.
Yeah, this was the presentation that we were going on. Yeah, so moving on now to we finished with the next train. So now we are moving on to this particular site where it shows that there were about, on 8 September, there were about 142 vaccine candidates in preclinical trials, about 29 in phase 1, 18 in phase 2, and about 9 in phase 3. And the candidates which were approved. So the real thing was, when there was a pandemic and the vaccine is available, at that particular time, the rising nationalism and the fraying global cooperation, you can say, how can we ensure rich countries don't stockpile the shots, leaving the poor countries out in the cold? So a plan came up by the WHO to ensure that there is fair access to the potential vaccine, and this was called as COVAX. So COVAX is one of the 3 pillars to the access to COVID-19 tools accelerator, which was launched in April and by the WHO, and it was coordinated by Gavi, that is Global Alliance for Vaccine and Immunization, and it was also partnered by CEPI, that is Coalition of Epidemic Preparedness Innovation, and the WHO together. So it was a platform that will support research, development, and manufacturing to the wide range of vaccine candidates, negotiate the price, and all the countries will participate in it equally, and there will be equitable access to the diagnostics as well as treatments and vaccines. So whatever companies we are going to see today, which are the vaccine candidates, are all entered into an agreement with COVAX that they will share all their vaccine details and the vaccines programs to all the poor countries also. So this was one. And the other thing which was to be seen is all the vaccine candidates that you're going to see today are also EAU, that is it has received authorization for the use. And these are the few papers which show that the vaccine deep drive, that is all the candidates which started on with the vaccination program, how they process. Normally, when vaccines, medicines, and medical devices go through various rounds of testing before being coming into the final regulatory approvals of the country, But during healthcare emergency, this particular process is shortened and the criteria is made simpler. And this is what happened during this pandemic. And we have reports here how they started their work: the Pfizer, Moderna, AstraZeneca, Covishield, Covaxin, the Novavax, and Sputnik. And in order to gain entry, to start with the basics, right in the center you have a diagram. In order to gain entry for the virus into the cell, there is these are the enveloped viruses which use proteins or glycoproteins to fuse their own membranes to that of the cells. These are the functional units which bind to the protein on the surface of our cells called as ACE2. This is the basic, triggering uptake of the virus particle and eventually the membrane fusion. Given how crucial the spike protein is to the virus, many antiviral vaccines or drugs are targeted to the viral glycoproteins. The New England Journal, which you see on the left-hand side, July 2020, gives a preliminary report of the mRNA used for vaccine preparation. The Moderna and the Pfizer vaccines deliver strands of genetic material known as mRNA, give instructions to our immune system to make our own version of protective antibody and the T cell production. While the article, you can see the Lancet below on the left-hand side, it's December 2020, a case study of safety and efficacy coming from Brazil, South Africa, and UK. The AstraZeneca vaccine uses a modified cold virus found in chimpanzee, and it's an adenovirus. It's weakened, and so it does not cause any illness in people. The virus delivers the spike protein's genetic material. Again, you can see the next one, the New England Journal, came up again on the right-hand side. It gives a safety and efficacy report on the trials with mRNA 123. Then the status of the immunological memory after 6 months of infection of SARS-CoV-2 was reported in November 2020. All of these candidate vaccines work by presenting their spike protein to the immune system. The spike proteins aren't dangerous because the rest of the virus isn't present. However, the body now sees the protein and designs an immune soldiers to fight it upon future exposure. That is the basic understanding of the vaccine. So now coming to the industries involved in the vaccine production, all these, as I said, have received their EAU. That means everyone who has the vaccine injected has to be followed up, tracked, and monitored. That is what is called as emergency use. The note, and also there is one note there given for all the scientists who they warn that directly comparing and the efficacy of the vaccine is not allowed because each trial was run differently. So now what we are going to see is we're not going to compare the vaccine candidates, but we are going to go through the key features so that in the next week when we have the next lecture, we understand the technology of the virus. Okay, so this is a typical virus candidate which directly stimulates the immune response. A microbe, traditionally what you see is a microbe or a protein injection sets off alarms, inflammation occurs, and antibodies are produced. The same eventually happens with mRNA vaccine also, but there is a crucially different first step. The mRNA must be taken up by the body, and then the own body cells produce the protein that stimulates an immune response. Pfizer was the first to report its clinical trial, and it is the company is called as Pfizer BioNTech COVID vaccine. It's the code named by BNT 162b2, and it is also named as Togemeran, which is sold under the brand name of Comirnaty. So COMIRNAT, Comirnaty. Okay, so Pfizer and German biotechnology company called as BioNTech developed this particular Togemeran. And it is nothing else but a nucleoside modified mRNA, which is also called as mod RNA. And this vaccine is nothing else but it's encoding an optimized full length version of severe acute respiratory tract virus spike proteins. The name Comirnaty is coined from COVID-19, and you can find here that it starts with COM, that is community. The middle word is RNA, and it ends up with TY, that is immunity. So it's immunity for the community. That was the name given. And this particular virus company, you can say the technology is from the BioNTech. The BioNTech company is a small German-based company, and started by a Turkish husband and wife with the basic technology of the vaccine. And Pfizer became a platform. It is partnering with Pfizer, a giant company that produces products in the market. Of course, Pfizer has many products in the market. It's a unique company. And mRNA, it uses mRNA. So these vaccines are used for the first time, developed for the first time. It's a unique example of how technology could move in manufacturing too fast. And the antigen used, if you see the table here, the antigen used is spike protein, the preservative. There are no preservatives, no adjuvants used. There is no DNA change, no allergic reactions, few, but they are not related to the virus. Efficacy, much to everyone's surprise, the experimental mRNA vaccines are most effective, 95%. Storage temperature, Pfizer vaccine poses a logistic problem here. It has a storage temperature of minus 94. Cold storage chain is required here, special. It requires a deep freezer. And it has a interval dose of about two doses two doses by an interval of 21 days. And it has that particular regime, 0.5 ml, 1 month apart. Most vaccine at this stage require 2 stages, two doses, all of them except the one, which I can tell you later. The mod RNA is formulated in lipid nanoparticle for administration via intramuscular injection in the deltoid area. It is at the age of 16 and above given. There is no infertility issue. The infections as seen for in the clinical trials were only 8 out of 162. No severe deaths due to COVID after infection, after vaccination was none. Then the transmission, you can say it's unknown. Stops long hauling symptoms, do not cause long hauling. Nothing is known here, and long time efficacy or side effects are also not known. So this is the company which got the first license and started the first manufacturing. Coming to the next one equally is Moderna. Moderna is into RNA viruses or RNA, RNA utilization for a very long time. It's US-based small company, Cambridge. Its candidate vaccine is called as mRNA-1273, which was developed in collaboration with the US government. The company size does not have any products in the market as yet. They are all in the trials, phase 2, phase 3 trials. And so far, none of them have been come out. So Moderna uses experimental technology that again employs mRNA. This is called as mRNA technology platform. It is published in Nature, August 2020, and they received an EAU on December 19, 2020. Do they have any products in the market? Moderna does not have any products in the market. They have many RNA-based vaccines, all in trials, as I said, and still to be approved by FDA and WHO. Antigen use, spike protein, no preservatives. Antibodies, no. Adjuvants, no. Do they change DNA? No. Which antigen for the virus do they make? Yeah, they make spike proteins. They all make spike proteins for the virus. So far for Moderna, no allergic reactions have been occurred. Efficacy is 94%. Storage is just normal, minus 20. So they can stay for months in a refrigerator. Then each 0.5 ml dose of the Moderna COVID vaccine contains 100 micrograms of nucleoside modified mRNA, encoding the spiral protein, glycoprotein of SARS-CoV-2. Interval, the dosing regime is 2 doses, 0.5 ml, as I said, 1 month apart. Most of the vaccines at this stage require 2 doses. And the schedule is 28 days. Administered intramuscularly in the deltoid region. Then there are, in the vial that you get multi-dose, that is the age indication is 18 years or of age or above. No infertility, no infections are 5 out of 90 cases, no severe ones, and the deaths are 0. The trials are shown in their reports into the test group, as well as the placebo group, as well as the blind studies. Do they stop transmission? It's unknown as yet. Going to the next one is the Astrazeneca. Yeah, Astrazeneca uses it was formerly called as AZD1222, was co-invented by the University of Oxford, and its spin-out company called as Vaccination. Vaccination is a clinical stage biopharmaceutical company. It's linked to the Oxford's Jenner Institute, one of the oldest and most renowned vaccine research center in the world. The company has also formed around 2 viral vaccines, one to prevent influenza and the other to treat prostate cancer. AstraZeneca collaborated with Oxford University. So the technology is from Oxford University and the platform for manufacturing is AstraZeneca. And they have changed the code name. It is ChAd-OX1 COVID-19 coronavirus vaccine. It is a recombinant vaccine. This is a very different technology than the Pfizer and Moderna. And the name, as it says, ChAd, that is CH is chimpanzee, AD is adenovirus. It's Oxford version 1, so it is OX1. So this is built scientifically by the Oxford College. The carrier virus is adenovirus. It's a vector virus used in chimpanzees, adenovirus that has been genetically changed so that it is impossible for it to grow in humans. The adenovirus from chimpanzee is used. It is also called as replication deficient chimpanzee viral vector, which is a weakened version of a common cold virus, adenovirus, that causes infection in chimpanzees. It contains the genetic material of the SARS-CoV-2 virus spike protein. After vaccination, the surface spike protein is produced, priming the immune system to attack the SARS-CoV-2 virus if it later infects the body. The antigen production is spike protein. The dosage is 2 doses, 1 month apart. The storage is 6 months. Again, the normal temperature, 2 to 8 degrees Celsius. Normal refrigeration. It highlights no hospitalization or severe cases in vaccine group or placebo group or even the blind studies. Another interesting thing is the storage. It can be stored for 6 months at refrigerated temperature. The trials were conducted in US, Japan, Russia, South Africa, Kenya, America, Brazil. The side effects with no serious side effects which are related to the virus. And the efficacy was 70%. Age range is 18 years. And the protection after 14 days after the second dose. So it's just a normal technology, time-tested technology that is used here, where adenovirus becomes the vector vehicle. The next one, India had approved 2 viral vaccines. One is the previous one that we have seen, AstraZeneca and Oxford University, and the other its own, that is Bharat Biotech. The Indian company Bharat Biotech partnered with National Institute of Virology and Indian Council of Medical Research to develop an inactivated coronavirus vaccine called Covaxin. India authorized the vaccine for emergency use on January 3rd, and it is the first vaccine that has been developed in India, got the clearance, and it's currently reporting the results of the 3rd vaccine. I think not only reporting, the reports have already come out 2 days back with the efficacy of 81%. And that's good news. Covaxin of Bharat Biotech contains whole inactivated virus. The code name is BBV152. The vaccine is developed using whole virion inactivated viro cell derived platform technology. Inactivated vaccines do not replicate and are therefore unlikely to revert and cause pathological effects in human beings. Conventionally, inactivated vaccines have been around for decades nearly. Numerous vaccines for diseases such as seasonal influenza, polio, rabies, Japanese are to date, and it is well established and a time-tested platform, I can say, in the world of vaccine technology. And Bharat Biotech has been doing it for a very long time. Covaxin is included along with immunopotentials, that is the vaccine adjuvants. It has an adjuvant here, which is alum precipitate, Algel, IMDG, or Algel, you can say. It is a 2-dose vaccination regime given 28 days apart. The temperature is the same. The storage temperature is no sub-zero temperature. So it's a normal refrigeration. And it's given in the multi-dose vials, stable at 2 to 8 degrees Celsius. Preclinical trials were performed, demonstrated strong immunogenicity and protective efficacy in animal challenge studies conducted in hamsters and non-human primates. The safety and immunogenicity of an inactivated SARS-CoV-2 vaccine, BBV-152, in a double-blind randomized phase 1 trial is already reported on January 21, 2021, and it has been already shown that the efficacy is quite well. But the third phase trial had to come out, and the results had to come out, and this came out 2 days back, and it shows 81% says Bharat Biotech reports. The phase 3 study enrolled 25,800 participants, 25,800 participants between the age group of 18 to 98 years of age, including 2,455 over the age of 60 and 4,500 with comorbidities also. So this is the vaccine which has come out and been produced in India solely. We have the next, the dark horse of the COVID vaccine race, that is Johnson & Johnson, J&J. J&J vaccine is actually the vaccine with a single dose, and it has standard fridge temperature storage. So these are the two important things which makes it different from the other vaccines. So the company is actually Janssen in collaboration with Johnson & Johnson. It is developed by Janssen, the Belgium-based arm of Johnson & Johnson. The vaccine name is AD26-CoV2.S. That is adenovirus name number 26, adenovirus 26, used for CoV, that is COVID vaccine 2, and the S is for spike protein. So this is the basic technology. The vaccine uses a harmless adenovirus to deliver genetic material that tricks the human body into producing proteins known as antigens that are normally found on the coronavirus surface, helping the immune system to develop an arsenal against the infection. That is given on their website very clearly. The Janssen investigational COVID-19 vaccine leverages the company's ADVAC vaccine platform. So the platform is ADVAX, that is adenovirus vaccine, which was used to develop and manufacture in Janssen's European Commission approved Ebola virus regime. And it also has a platform like for so many viruses, Zika virus, RSV, HIV. So this particular platform is quite old for Janssen's, and it has been manufactured by Johnson & Johnson. They have a particular cell line which they have proprietary rights, that is PERC6, in which they develop this particular adenovirus. And this particular cell line is also developed from a fetal cell. We are coming to it in our next lecture, how this technology is developed and how the vaccination is produced. The platform is, as I said, ADVAC, which they have been using it for years. So the technology is age old. This virus can be stored for 2 years at -20, at least 3 months when it is at the normal temperature, refrigeration temperature. Dosage is 5 × 10⁻10 virus particles. 0.5 ml is the size of the dosage per vial, and it is given intramuscularly. It is transmuted in normal fridge, then it is stored in a normal fridge and fridge temperature. The vaccine has been authorized emergency use for adults about 18, and it works under a study called as Assemble. It is a randomized, double-blind, placebo-controlled clinical trials studies, which shows 66% effectiveness in preventing COVID-19 in a large stage global trial. So the platform that the trial groups that they have used is in US and Latin America and South Africa, and they've got efficiency like 77, 72%, 66%, and 57% in South Africa. So the company has a data which has the last trial data also, and they entered into the COVAX program in December, and this vaccine also is you can say it's one-shot vaccine. That is one most important thing that has been given here. Okay. The next one, again, interesting one, is a Novavax. It has a beautiful, fantastic technology, I can say. And this is an American vaccine development company headquartered in Maryland. They have facilities in Uppsala, Sweden also. It's a biotechnology company that promotes improved health globally throughout discovery, development, and commercialization of innovative vaccines. So the COVAX COVID-19 vaccine, codenamed NVX, that is Novavax, and COV, that is COVID vaccine 2373, and also called as SARS-CoV-2-RS, that is recombinant spike protein nanotechnology they are using. That is a platform, and they are using adjuvant called as Matrix M1. So this is developed with along with the coalition, that is CEIP, Epidemic Preparedness Innovation. It is protein-based vaccine candidate engineered from genetic sequence of technology to generate antigen derived from coronavirus spike protein. They have an adjuvant, which is a saponin protein-based matrix M, to enhance the immune response and stimulate high levels of neutralizing antibodies. So it's stable at 2 to 8 degrees, a normal temperature. It requires 2 doses. In late January, Novavax electrified by announcing that the COVID-19 vaccine showed efficacy to about 89.3% in their first primary analysis of data. From a phase trial in UK, the variant, it also showed good efficacy towards with the UK variant B117. And this is achieved in the phase 3 trials. They have 60% of efficacy in South Africa, and it's known to have shown 90% on an average when they take it together from all the countries. So Novavax, as I said, has an interesting technology. I love this vaccine actually because of its technology. It's very different from the traditional attenuated vaccines. It is very different from the adenovirus vaccine or the mRNA vaccine of Moderna and Pfizer. What do they do here? They select the genetic sequence to encode the vaccine antigen. Genes are cloned into baculovirus, and this baculovirus infects the moth cells. They have a cell line, moth cell called as SF9. So they transfer it into the cell line. The antigens are expressed by this moth cell lines. The spike proteins are then harvested. They are purified as multimeric nanoparticles. So they are again enclosed the same way as the Pfizer and Moderna does. They are enclosed in a nanoparticle for the transfer of this particular virus material into our cell. So to promote superior immunogenicity and better functional immunity, they have said SF9, that is, and BV, insect cell platform. This efficiency expresses large antigens and particles. So this is a quite different technology than all the technologies that we have seen in the previous few slides. And it's a wonderful technology. We'll see it in the next week. The next one is the first registered COVID vaccine proven human adenoviral vector technology. That is a Sputnik vaccine. Sputnik V COVID-19 vaccine registered in August 2020. The company Gamaleya is research institute, state-backed company, research institute of immunology, you can say. It is the vaccine type, the name of the vaccine is GAM COVID Vac. The vaccine type is adenovirus ADV26/5. Now this is again a very interesting technology that they have used. They have used a human virus, not chimpanzee virus as in Astrazeneca, but it is adenovirus 26, which is a human adenovirus, same one that J&J is using. So they always say that we have the first part of J&J. It's always mentioned in the reports. That means that they are using the adenovirus 26, just like J&J has used. And then in the second dose, they are using adenovirus 5. So the 2 doses which they are injecting, which they have in the vials, the first dose is using a vector virus adenovirus 26, and the second dose they are using adenovirus vector, that is 5. So they have both separate vectors here. So antigen production, they have DNA full length for spike protein. None of the adjuvants are used. Adenovirus itself, this adenovirus particle itself is true enough to trigger the immune response. The Sputnik virus comes in 2 doses. This is the storage is minus 18 degrees Celsius, and they give it in a lyophilized form also, which can be stored at temperature +2 to +8 degrees Celsius, which allows for easy distribution worldwide, including the hard to reach remote areas. The dosage is 10 to the power of 10 virus particles, and the volume is 0.5 ml per dose. The vaccine that is using adenovirus 26 virgin is the first, and the other dose is the vaccine that is using adenovirus 5, triggering a little more immunity. This is an interesting virus. That is why they say the phase 3 interim results showed, interestingly, 91.6% efficacy. The trials are done in India, UAE, Venezuela, and Belarus. The Sputnik virus is one of the 3 vaccines in the world with this efficacy, and it is the first, and which shows the placebo as well as the final control points as 78 confirmed COVID-19 cases. Deaths are 3, which are unrelated. Out of the 16 got infected after the first dose of vaccination. The adverse results, none vaccine related. 45 participants showed mild signs and symptoms. So the safety and efficacy of an recombinant adenovirus 26 and recombinant adenovirus 5 vector-based heterologous prime boost, you can say, COVID-19 vaccine, which has an entry results shown in the Lancet in 2021 February of its 3rd trials. So it's a very interesting one. The next one is Sinopharm. It's again a China vaccine. China National Pharmaceutical Group Corporation company, referred to as Sinopharm, and it is state-owned enterprise founded in November 26, 1998. Headquarters is in Beijing, China. The name of the vaccine is BP CORV, that is, it is recombinant virus again used, developed by Beijing Institute of Biological Products. It's one of the 2 inactivated viruses that have been developed by Sinopharm. So it is an isolated from a patient, very interestingly, and they have grown it in the laboratory using cell lines. The virus is inactivated using a chemical called beta-propiolactone for inclusion into the vaccine. This is the adjuvant that they are using. The vaccine also includes an aluminum hydroxide vaccine adjuvant, as I said, which boosts up immunity. Aluminum hydroxide is used in many vaccines even previously or long back. It relies on traditional technology similar to, they always report it as it is similar to CoronaVac and BBV152 that is shown by Bharat Biotech. So CoronaVac, also known as Sinovac, it is using inactivated virus developed by this particular biotech company. And the BBV152, it's also known as Covaxin. It is again inactivated virus-based COVID-19 vaccine developed by Bharat Biotech in collaboration with ICMR. So one of the remarkable advantage of Sinopharm is it is a convenient storage transportation, which just needs under the environment of 2 to 8 degrees Celsius with no special and harsh requirement, so that can be transported well within, requires no different cold chain. 2-dose immunization, day 0 and 28 days are 2 days apart, and the doses are 2 microgram, 4 microgram, and 8 microgram. It's given in the 2 different age groups, 18 to 59 age, as they show in their reports. Induced neutralizing antibodies in 100% of vaccination recipient. Age approved, 18 years and above. 5 phase trials are there in Argentina, Bahrain, Egypt, Morocco, Pakistan, Peru, and UAE. Side effects, no serious side effects, no adverse effects. But the most important thing is this vaccine is very costly. If you compare the rates of vaccines, this is the most costliest one. Safety and immunogenicity of the inactivated SARS-CoV-2 BV1, BV, CoV-V, randomized, double-blind, placebo-controlled group in phase trial 1 and 2 are been reported in Lancet again. And they show that in the company there was 879.34% of efficacy, and UAE it was 86%. So the reports are already out. So the takeaway today is why biotechnology has to offer vaccine development, what it has to offer. New developments in biotechnology have led to a number of new approaches for the development of vaccines aimed at preventing infectious diseases. One of the important aspects to understand here of science which has emerged over the past century is the realization and importance of the vaccine construct, the construction of the vaccine, which involves two major components, the immunogen itself and the vaccine platform. The mutations, as we have seen, are a natural part of virus life cycle and occurs when a virus causes infection. Most mutations are useless or weaken the virus, but occasionally a mutation may benefit the virus. Variants arise in people, not the environment. Viruses are useless without a host cell. If we can prevent infection through vaccines, physical distancing, mask wearing, and hand washing, then we can prevent the rise of more variants. All our current vaccines are specifically designed to target the original form of the coronavirus that appeared in 2020. While it would be ideal for a vaccine to be an exact match for the circulating virus, this is unlikely to happen due to the mutations of the emerging variants seen nowadays. The good news is that vaccines don't have to be a perfect match against a virus to provide protection. Each mutation alters a small part of the virus. These are the latest mutants, and they are a little different from what seen before. But even the most genetically distinct mutant that are arising, they are still 95% of the or more identical to the original version of the spike, meaning that our immune systems have a really good shot at stopping the virus completely after vaccination. Eventually, scientists may need to update the vaccine to target emerging variants, but as for now, it is a race to get as many people protected as possible before the virus changes again. As more people are vaccinated, fewer will become infected, and the virus will have nowhere left to go. That shows how we win. Each country should take care. This is a global pandemic requiring a global response. No outbreak is in any part of the world it's an outbreak. An outbreak in any part of the world is an outbreak in the entire world, and no countries will be safe unless all of us are protected. That is what we have seen today, all the vaccine candidates. Now, next week, we will see how these technologies are based, what is the proper processing of these mRNA or the adenovirus technologies that these companies are using. And in the third session of my series, we will see the other techniques that are used by all the biopharma companies. So talk to you all next week. Thank you so much.
Thank you very much, ma'am.
Good night to you all. Yes, most welcome.
Thank you.
Thank you, ma'am.
Most
Thank
welcome.
you. It was very, very useful.
Yeah.
Yeah.
You all are
Very
most welcome.
Yeah. Full of knowledge. Thanks a lot.
Yeah, I thought when we are seeing the big trends, let us start with the recent trend, that is vaccinology. So I started with it. And as I'm dealing with biotechnologies in industries, I said, let us see the industries, what they are doing, and then the biotechnologies, and then the other technologies which they are using. Yeah.
It was most interesting in the present scenario.
Thank you so much. Yeah. It is definitely.
Very informative talk, Dr. Sarita.
Yeah, thank you.
Yeah, great talk, Dr. Sarita. Thank you so much. So I have a question. Can I ask one?
Yeah, definitely.
So,
Go ahead.
This Sinopharm Chinese vaccine.
Yeah.
It's an inactivated killed vaccine by beta
Yes. Yes.
So in what cells were they maintaining this? Because that is not shown here.
Yeah, most of the reports, Yeah, most of the reports of China vaccines are not very clear. My
Yes.
today's talk was based exclusively on the reports that I get on internet. So after, there's one more vaccine coming up in China, but I did not get lots of it. I did not include here that particular vaccine. But of course, as you say, that it's not very clear how they are doing it. The procedure is not very clear, and whatever I could put my hands on with the published papers, I tried to bring it to you.
No, even I tried to find out, but I couldn't find what its cells are they maintaining this virus in.
Yeah.
Okay.
But I think eventually they'll publish. I think some papers will come up now.
Yeah. But that's the secret. I don't know.
Yeah.
Okay. Thank you very much.
Great
Most
talk,
welcome.
Dr. Sarita. Just one quick question. If you are given an option of using Sinopharm or Moderna or mRNA-based technology, which option for vaccination would be the best?
Yeah, that's a very good question. Most of the people ask that question, but I'm just waiting for the Novavax to come. I love Novavax technology. It's a beautiful technology. Yeah, I will get to it in detail in the next week, how it is built up, and I have read complete the technology on its website as well as the others. But it's a really fascinating technology, and it is very, very different from the other ones that are used. So I would, if given a choice, I would go for Novavax. Yes.
That is a VLP based, VLP novel vector based technology.
Yes. Yeah.
Perfect. Thank you so
Is
much.
it correct, what I have heard, that mRNA vaccines will create long-lasting immunity as compared with the protein or spike vaccines? Is it correct?
So all are using spike proteins, and we have to see now, because as the vaccines are being injected and everything has been traced and monitored, the antibody level has to be seen for all the vaccine programs, and there is not much time before we have got to see how this works. But I think in 1 or 2 years it will be quite clear how much the antibodies stay in our body and how much they are. We don't even know the threshold as yet, because even that things have not yet come up. Yeah.
That we don't know. We don't have got a time.
We don't have the data. It will come up eventually. This vaccine, yes.
Thank you very much, madam.
Yeah, welcome, most welcome.
Myself is Shilpa Jani. On behalf of Microbiologists Society of India and all 33 participating budding scientists of this webinar series on impact of biotechnology, I thank Madam Dr. Sarita Mahajani for enriching us all on current trends in the world of vaccination. We are all nourished by advanced and current knowledge on COVID-19 vaccine, ma'am. I also congratulate Jai Jatia for organizing such a nice event and keeping our knowledge updated. Thank you, madam. Thank you very much. It was very interesting to hear you.